Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Neisseria gonorrhoeae, often referred to as NN. Gonorrhoeae is a gram-negative oval-shaped bacterium that infects humans and causes various infections, including gonorrhea. The name Neisseria is derived from Albert Neisser, a German physician who discovered the bacterium, while gonorrhea originates from the Greek words gonos, meaning seed, and rhou, meaning flow, together translating to flow of seed, a description referring to the penile purulent discharge, which was mistakenly believed to be semen in infected males. Now, let's explore a bit of microbe anatomy and physiology. Neisseria gonorrhoeae is a gram-negative bacterium because its cell wall contains a thin peptidoglycan layer, which prevents it from retaining the purple dye used during gram staining. Instead, like other gram-negative bacteria, N. gonorrhoeae takes up the pink safranin dye. N. gonorrhoeae typically exists in pairs called diplococci, arranged side by side, giving them a coffee bean-like appearance. They are also non-motile, non-spore-forming, and obligate aerobes, meaning they strictly require oxygen to grow. Finally, they are catalase and oxidase positive, which indicates they produce both of these enzymes. N. gonorrhoea grows on a special chocolate medium known as Thayer-Martin agar, which primarily contains sheep blood. Certain antimicrobials, such as vancomycin and nystatin, are typically added to the Thayer-Martin agar to suppress the growth of unwanted bacteria, or fungi, thereby enhancing the selective growth of Neisseria species. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. However, other Neisseria species, such as N. meningitis, share similar properties. So, the maltose fermentation test is used to distinguish between the two. The key point is that Neisseria gonorrhoeae cannot ferment maltose, while Neisseria meningitis can. To perform this test, a pure sample from the suspected bacterial culture is transferred into a sterile tube containing phenol red maltose broth, which is then incubated at 36 degrees Celsius for 24 hours. Since N. gonorrhoeae doesn't ferment maltose, the solution remains red, whereas with N. meningitis, fermentation byproducts cause the solution to turn yellow. Now, unlike its sister species, Neisseria meningitis, Neisseria gonorrhoeae is not an encapsulated bacterium, meaning it lacks a polysaccharide capsule. However, this bacterium possesses a wide array of other virulence factors, which it uses to invade and damage host cells, as well as to evade the immune system. First, N. gonorrhoeae has pili, tiny thread-like extensions that radiate from the surface of the bacterium. These pili help N. gonorrhoeae adhere to the mucosal surfaces of the host. They also enable bacteria to physically connect with one another, forming what's called a conjugation pilus, a small, hollow rod through which bacteria can exchange genetic information, including antibiotic resistance genes. Interestingly, N. gonorrhea pili are composed of antigenic proteins that can change with each infection, a phenomenon known as phase variation. Normally, when a particular bacterium causes an infection, the immune system retains a memory of that bacterium's antigen configuration. So, If the same bacterium infects again, the immune system can recognize it and rapidly produce specific antibodies against it. However, since N. gonorrhoeae alters the antigens on its pili with every new infection, the immune system is unable to mount a swift and targeted immune response. Phase variation is also the main reason why there is no effective vaccine against Neisseria gonorrhoeae. Pili aside, other virulence factors of N. gonorrhoeae include toxins. A key one is IgA protease, a toxic protein that this bacterium uses to break down immunoglobulin A, IgA. IgA is an immune protein typically found in mucosal secretions, such as those from the vagina or cervix. It plays a role in opsonization of bacteria, essentially tagging them so that neutrophils can recognize and eliminate them. So, IgA protease disables this first line of mucosal defense. However, not all IgA molecules are neutralized, meaning some N. gonorrhoeae bacteria still get opsonized and are subsequently targeted by neutrophils. Inside the neutrophil, N. gonorrhoeae is enclosed in a phagosome, a bubble where reactive oxygen species, like H2O2, are released to destroy it. However, N. gonorrhoeae produces catalase, an enzyme that breaks down H2O2. Unfortunately, this gives N. gonorrhoeae the upper hand, allowing it to survive inside the neutrophil and hijack its energy resources to multiply. Eventually, the neutrophil becomes overloaded, bursts open, and releases a large number of bacteria into the bloodstream, a condition known as gonococcemia. Inside the blood, N. gonorrhoeae can deploy additional virulence factors. One of them is a cell wall antigen called lipo-oligosaccharide, or LOS, which is notorious for triggering a widespread immune response that leads to sepsis, where blood vessels dilate, blood pressure drops, and vital organs receive insufficient blood supply. And one final virulence factor of N. gonorrhoeae is its ability to perform sialylation, a process in which N. gonorrhoeae coats its LOS cell wall with sialic acid, the same molecule naturally present on the host's cells. This allows the bacterium to mask its LOS antigen, essentially making itself invisible to the host's immune system, like a form of camouflage. As if that weren't enough, N. gonorrhoeae can also spread from the bloodstream to other parts of the body, such as the joints or the heart. Most frequently, however, N. gonorrhoeae causes gonorrhea, a sexually transmitted infection. In males, gonorrhea presents as urethritis, which is inflammation of the urethra, but it can also spread to the prostate, leading to prostatitis, or to the epididymis, resulting in epididymitis. In females, N. gonorrhoeae can also cause urethritis, but more commonly, it leads to vaginitis and cervicitis, inflammation of the vagina and cervix, respectively. Through the cervix, N. gonorrhoeae can ascend to the uterus, fallopian tubes, and sometimes even the ovaries, resulting in pelvic inflammatory disease, or PID. Finally, PID can lead to a complication known as Fitzhugh-Curtis syndrome, which occurs when the inflammation extends to the peritoneum and then reaches Glison's capsule, the outer covering of the liver. This results in the formation of violin string adhesions, thin strands of scar tissue that connect the liver to the peritoneum. If N. gonorrhoeae infects a pregnant woman, it can be transmitted to the baby during vaginal delivery, leading to early neonatal conjunctivitis, a form of conjunctivitis that appears in the newborn between 2 to 5 days after birth. Gonorrhoeae can also lead to some rare but serious infections, often as a result of gonococcemia. When the infection reaches the joints, it can cause gonococcal arthritis, which is more commonly seen in sexually active adolescents. If it spreads to the heart, it can affect the heart valves, resulting in endocarditis. The first symptoms of a Neisseria gonorrhoeae infection are typically associated with genital involvement. In men, symptoms may include a burning sensation during urination and a clear urethral discharge that can become purulent and even bloody. In women, there is often a thick, white, purulent discharge from the vagina or urethra, which may also occasionally become bloody. If the infection advances to pelvic inflammatory disease, PID, symptoms can include lower abdominal pain and fever. Alternatively, in cases of neonatal conjunctivitis, symptoms may include swollen eyelids accompanied by mucus and pus discharge from the eyes. In gonococcal arthritis, multiple joints become inflamed, leading to painful swelling in areas such as the wrists, ankles, and elbows. With gonococcal endocarditis, symptoms may include fever, chills, sweating, and a general feeling of malaise. Diagnosis is usually carried out using a vaginal or urethral swab, which is then smeared onto a slide for biochemical tests, and gram staining, revealing pink, coffee bean-shaped bacteria inside neutrophils. Culturing the bacteria on Thayer-Martin agar is required for confirmation, but nucleic acid amplification testing, NAAT, can also be performed. This method identifies the bacterial genetic material. Treatment for gonorrhoeae infections involves third-generation cephalosporins, typically ceftriaxone. However, gonococcal infections are often found to be associated with chlamydia trachomatis co-infection. Therefore, azithromycin or doxycycline is usually administered alongside ceftriaxone to also treat chlamydia. Finally, since gonorrhea is a sexually transmitted infection, it can be prevented by using condoms during sexual activity. All right, as a quick recap, Neisseria gonorrhoeae is a gram-negative diplococcus that grows best on Thayer-Martin agar. It's non-motile, non-spore-forming, oxidase positive, and catalase positive, but maltose negative. It lacks a capsule, but possesses several other virulence factors, including pili and proteins like pili, OPA, and AGA protease. Most commonly, gonorrhoeae causes gonorrhea, which presents as urethritis in males and vaginitis and cervicitis in females. If left untreated, infections can progress to gonococcemia, leading to serious complications such as gonococcal sepsis, septic arthritis, and endocarditis. Treatment involves ceftriaxone with azithromycin or doxycycline added to address a potential chlamydia trachomatis co-infection.